Hello, my name is Maham Farooq and I'm a student from the Brentwood High School, and I will be doing my presentation on the behavioral effects of acute sodium fluoride exposure on sensory and motor function in Drosophila melanogaster. Sodium fluoride is a substance that is so common, it's in the water you drink every day and the toothpaste you use every morning. For decades, we have tried to view sodium fluoride through the lens of dental health as a protector, but what happens when we look past the teeth and more into the brain? My research seeks to uncover whether short-term spikes in fluoride exposure can actually alter the fundamental way an organism interacts with its environment. So the danger of fluoride lies within its dosage. While it strengthens the bone at high levels, it becomes a neurotoxin at one point. It creates oxidative stress and disrupts the battery of the neuron, which is the Na plus K plus ATPase pump. This is especially concerning for the millions of Americans who use private wells today, where fluoride levels are often unregulated and can reach double the recommended limit. Recent studies have linked these levels to lower IQ in children, suggesting that our nervous system may be more sensitive to fluoride than we previously thought. Before getting into my experiment, I wanted to explain why fluoride exposure matters outside of the lab. Fluoride exists on a spectrum. Too little can increase cavities, and too much can weaken your teeth and bones and even cause fluorosis. More recent, some studies have raised questions about possible neurological effects at higher exposure levels. This is especially relevant for people who rely on private water. As you can see in chart number 2, about 15% of the US population relies on private wells. And that is especially concerning because those wells are not regulated for fluoride levels. Exposure can vary so much, understanding even short-term neurological effects becomes important. This gap of knowledge directly led to my research question. I wanted to know, does acute exposure to sodium fluoride specifically target motor coordination, or does it interfere with how an organism processes sensory input? I hypothesized that we would see a measurable decline in complex behaviors, but I needed a reliable way to test this without the ethical or temporal constraints of human subjects. This brought me to a very small but powerful research partner, which were the fruit flies. We use sodium fluoride every day for our teeth, but what it does to our brains is the short term question that is still unanswered in the scientific world. The fruit fly Drosophila melanogaster is a biological powerhouse. They share about 75% of human disease causing genes and have a central nervous system that mirrors our own in many ways. By using these flies, I could observe complex behaviors like climbing and light seeking in a controlled 24-hour window. Once I had my model, I designed a two-part testing system to separate physical ability from sensory perception. So the goal here was simple. If we expose these flies to fluoride for just one day, 24 hours, can we actually see their behavior change? More importantly, does it hit their physical strength first or their ability to process the world around them? So to separate these flies by gender and to put them into these vials, I had to first put them under NutriFly anesthesia. And within 30 to 45 seconds, they all fell asleep, and I was able to put them under the microscope and separate them by gender. I used three groups, a control, a 20, and a 100 mgl group. And after 24 hours of exposure, I ran two tests. The first was the negative geotaxis, where I tapped on the flies onto the counter and timed their climb up to measure motor strength. The fluoride was delivered through NutriFly medium over a 24-hour period, and each vial contained 20 flies with 5 replicates per group, and all environmental conditions were carefully controlled. The design allowed me to isolate fluoride concentration as the independent variable. To measure the motor function, I used a negative geotaxis assay. Flies naturally climb upward after being tapped down, and I measured their climbing success with a 20-second window. To measure sensory function, I used a phototaxis assay, which tests how strongly flies move toward the light. Fruit flies are naturally attracted to light, so a reduced response suggests that sensory impairment rather than just lack of motivation. So interestingly, the motor function data showed no significant change. Whether the flies were in a control group or the high exposure group, their ability to climb remained steady. Physically, the flies were still strong and their muscles were functioning normally. This suggests that acute sodium fluoride exposure does not impair motor function within a 24-hour frame. So when we look at the sensory data, the impact of fluoride becomes undeniable. We saw a significant dose-dependent decline in light response. The phototaxis index dropped from 0.49 to 0.25 in the high exposure group, with a p-value of 0.002. This confirms that while the flies could move, their sensory drive and ability to process environmental light were being compromised. So, What does this distinction between motor and sensory health tell us about the risks of fluoride? In conclusion, the acute sodium fluoride exposure selectively targets sensory pathways before it affects motor function. This suggests that sensory disruption is an early warning sign of neurotoxicity. This research highlights that even a short 24-hour spike in fluoride levels, which is common in unregulated water sources, can have immediate effects on neurological processing. While these findings are significant, they open the door to even deeper questions for the future. So, for future research, I hope to look at the cellular level, specifically whether this sensory decline is caused by oxidative stress or a disruption in neurotransmitters like dopamine. Future research could expand on these findings by examining longer exposure durations to determine whether motor deficiencies eventually emerge. Biochemical assay measuring reactive oxygen species or neurotransmitter levels could help connect behavioral changes to underlying mechanisms. Additionally, recovery studies and multi generational experiments could provide insight into whether these effects are reversible or persistent. So, thank you so much to my mentors at Brentwood High School for helping me and guiding me through this. And these are my works cited. Thank you so much.